This is a recording of the technical seminar delivered as part of the 2025 Roscoe Ellis Jr. Lectureship in Soil Science at Kansas State University, presented by Dr. Alexandra Kravchenko, a professor of soil science in the Department of Plant, Soil, and Microbial Sciences at Michigan State University.
We're so glad to have Dr. Sasha Kravchenko here. It's been a great visit. We've enjoyed having her and had a great Ellis lecture last night. I guess for those of you who haven't had a chance to interact with her or weren't there at the Ellis lecture last night, she's here from Michigan State University, soil scientist, studying all sorts of areas in soil science.
Yeah.
Biogeochemistry, physics, spatial soils mapping, all sorts of things, but has done a fair amount recently on looking at nanopores and carbon in those pores and the environments. Anyway, so this morning we're glad to have her present a presentation titled Below Ground Interplant Carbon and Nitrogen Exchanges and Their Contributions to Plant-Derived Carbon Soil Inputs. Thank you for coming.
Thank you very much for, yeah. So I don't cover. Thank you very much for the invitation and for introduction, and thank you for being here, listening to me second day in a row. So yeah, as that's the title. So what? Okay. Let's see. So as you might have guessed from my yesterday's talk, my big area of interest is soil carbon and its relationship with soil structure, that is specifically soil pores. So those two are really interconnected, and gains in soil carbon means improvements in soil structure and better development of soil pores. And then better development of structure means greater protection and greater storage of soil carbon. But both of those, they're driven by plants in essence. Well, not solely, but to a big extent. So plants are the source of the carbon inputs as rhizodeposits, root exudates, plant residues that are the biggest source of carbon in the soil. And also plants are very substantial driver in affecting formation of soil structure and formation of soil pores. So with that in mind, what was the thinking that drove this study is the role of plant community composition or plant diversity, diversity of plant communities in terms of their effects on soil carbon and soil structure and their interaction. So it is very well documented and very well known that diverse plant communities, that they are very good ways to increase soil carbon, that they can do it rapidly, they can do it efficiently, they can lead to stable accrual of new carbon in the soil. And there are a variety of mechanisms through which this positive effect of diverse plant communities acts. So what I'm going to talk about today is what we hypothesize is just one of many of these mechanisms through which diverse plant communities might make their positive contribution. Specifically, these mechanisms that we hypothesize and explore what role it actually might play is interplant carbon exchange below ground. So our hypothesis is that it matters. So what is interplant carbon exchange, or what is interplant carbon transfer, is when a carbon that is photosynthetically procured by one plant when it somehow, through some below ground mechanisms, ends up to be part of the body of a neighboring plant. So one plant gives its carbon away to a different plant. It is not a new phenomenon, newly found phenomenon. It has been known for like 60+ years. And there has been quite a few works studying it that demonstrated that not always, but at times, this carbon transfer can be very fast, and it can be very sizable. Also, there were studies that demonstrated that it is possibly that distantly related plant species might be actually more prone to share their carbon than the closely related plant species, which is one of the sort of reasoning why we think it might be a contributor to diverse plant communities' contributions. There are two major ways in which this carbon exchange can take place. One would be a direct contact between the roots, that either two roots completely close to each other and carbon goes from one to the next one, or just deposits generated by one root at some point in time are encountered by roots of another plant at other point of time. That would be one way. Another way would be through direct links by fungal hyphae through AMF fungi, for example, connecting the roots of different plants. So how all this would be in, like what we were thinking, why that would matter for soil carbon gains is the hypothesis is like this, that the more carbon is transferred from one plant to the other by whichever mechanism, the more of it will be lost in the soil, and the more of it will then contribute to soil carbon gains. And again, it could be that the more diverse plant systems, that they might be more effective in exchanging carbon among their community members, and as a result, there might be more exchange, there might be more why they might be depositing more carbon below ground. So with that in mind, one, sort of a big practical question related to that is, as we are thinking of the best ways to develop those diverse plant communities, if we have in mind the idea, how do we build the ones that are most effective in bringing about carbon gains, it might be good to know what exactly to look for. So should we be just aiming at highly diverse plant community, just regardless of what plant species are there, just make it mixture of very diverse species and it will work? Or could it be that certain presence of certain species or species representing certain functional groups, maybe that actually would be more effective way of doing things? But of course, to answer these questions, and I'm not going to have answers today, it's like a big vision of where we want to go with this research eventually, not right now, we're not there yet. But having the answers would give us a way to decide as what would be the best way to build diverse plant communities most effective in leading to soil carbon gains. And so the witchgrass, it happened to turn a great model plant explore these questions. So okay, so first of all, I should make sure I tell you why switchgrass, that's why we were so focused on switchgrass, is because it's currently regarded as a high potential bioenergy crop, a feedstock source for bioenergy and bioproducts creation, that it has a lot of benefits from that perspective. It has very large above ground biomass that can be harvested for that bioenergy, bioproduct production. It is native, so not some exotic plants and not something that will turn into invasive species. It's easy to work with. It's very low nutrient requirements for it. It can go all over the country, starting from northern parts like Wisconsin, Michigan, all the way to south and east and west. So from that perspective, it's like very promising bioenergy feedstock plant. And so that's a lot of studies devoted to that. But besides all that practical importance of switchgrass, we found it to be, as I was saying, a very interesting plant to work with from perspective of this soil carbon gains and role of plant community composition on it. Why is that so? Because as our work and work of many other studies in different locations across the country, what people demonstrating is that monoculture switchgrass, when it's just a stand of switchgrass itself, it's very slow to generate soil carbon gains. It's very surprising. No one could believe it first when they saw it, because it just made no sense whatsoever, because switchgrass has this giant root system. So how could it be that it doesn't increase soil carbon very rapidly? Well, it doesn't. So what you see here is just one example of one of the experiments where it's been demonstrated. So here, these are soil carbon data after 10 years of these systems, and systems going across this gradient of plant diversity from continuous corn to that monoculture switchgrass, and then continuous increase to 24 plus species here. And you can see that the very, again, 10 years, this giant switchgrass with giant roots there, and the soil organic carbon concentration is just as the same as this continuous maize. But as soon as other species are being added, like here switchgrass is mixed with Canadian rye, and even just, I don't know what I'm showing was, maybe I should use that.
Do you want a pointer?
Yeah. So like when just one thing, Canadian rye, was added to the switchgrass in the two species mixture, here is the carbon goes up, and then.
Do you want to plug this in to your computer to advance and move your slides or not?
Maybe not, I ran out of.
You just want to use that as a pointer?
Yeah.
Okay.
So we add just one more species and things change substantially. And of course, the most diverse system has the highest carbon to it. So when alone switchgrass doesn't increase carbon, when what other studies showed from long-term experiments with that looked at plant diversities in similar plant communities, is that actually when switchgrass is part of that plant community, then it contributes positively to soil carbon changes. So Here are the results of multiple regression from that work that looked at specific contributions of individual community members to soil carbon storage. And here it is, switchgrass with significant positive contribution. So by itself, it's no good. When it's a member of community, it's actually, doing positive things as far as contributing to soil carbon gains. So what we were thinking is that maybe these presence of these carbon exchanges, maybe that might be one of the reasons of why this, puzzling behavior of switchgrass in its contribution to carbon. And I can tell you right away that, this work actually didn't lead to conclusive answers, so we're still not sure. So it doesn't seem that carbon exchange is actually the answer to this particular question, but it was the motivating reasoning for why we started with what we started with. So what we did is two big greenhouses and a few small studies where we grew switchgrass with neighbors. And we used 13C labeling to track the 13C that would be originating from one of the plants, like switchgrass, and then we would see where it goes, where that 13C goes to different plants. So one of those studies was led by Dr. Zhang, who was a visiting scholar in our lab at the time. So what you see here is like our labeling chambers. And it looks like we have some trash bags right there covering the plants. That's exactly what they were, trash bags. And how this whole thing worked is we would have pots with multiple plants growing in them. So one of those plants would be open during the labeling. So it would have access to the CO2, 13C CO2 inside the chamber, and it would be doing photosynthesing at the time, taking it up. But the neighboring plants would be covered with those trash bags, black ones that would block the sunlight. They are not doing photosynthesis, so they are not taking up 13 CO2. So the only 13 CO2 that would be, if we find any in them, that would have to be originated from this uncovered source plant. And we looked at different combinations, like here would be switchgrass grown together with wild bergamot. In one of the pots, switchgrass would be open and it would be getting 13C. In other pots, a wild bergamot would be the open one, and that will be getting a 13C. So I'm not going to go in a lot of detail on this. I just want to show you what are the combinations of neighbors that we did try out in these multiple experiments. So these four are the source plants that we explored, and the list is the recipient neighbors that we explored, and the size of the arrows reflects very relatively the magnitude of that carbon transfer that we observed. So as you can see, the bottom line here is that there is a lot of variability within the plant community of who gives their carbon to whom and who doesn't. Some of the sort of standing out, interesting, consistent results that we observed is that switchgrass was very nicely exchanging carbon with big bluestem, another prairie grass, and that was sort of reciprocal. One would give carbon to the other and back. And there was lots of carbon exchange between switchgrass and bush clover. Some other things that also were consistent, black-eyed Susan, this lovely yellow flower. It also happens to be a very aggressive neighbor. So out of those two plants that we looked at, no one liked it enough to give it a lot of, to give it carbon. Wild bergamot seemed to be a very strange one also in this respect, because we did find it to be a recipient of carbon from other plants, but it seems it really wasn't interested in sharing its own carbon with anybody, at least anybody we tried. So, okay, so what is that makes, so there are probably a lot of reasons for this variety of results of who gives carbon to whom. One very straightforward one that we thought, well, we better start with is nitrogen availability. And here, this is that second greenhouse study we did that Jinho Lee, my former PhD student, did. Here, instead of pots, we used these big rhizoboxes, and the source plant, the labeled plant, would be in the center, and then we would have the non-labeled neighbors on the side. So specifically to answer this nitrogen availability question, what Jinho had is this 3 specific treatments in that overall giant experiment. In one treatment, switchgrass was, well, actually in the study, switchgrass was the focal labeled plant in most of the treatments. So in one of them, black-eyed Susan was on both sides. The other treatment, black-eyed Susan on one side and bush clover on the other. And one more treatment where, again, black-eyed Susan was on both sides. But what we did to one of those sides, we did this root split sort of nitrogen addition experiment with treatment with one of the sides, where we would basically fertilize or enrich this black-eyed Susan plant with nitrogen. And of course, clover would be naturally enriched in nitrogen. So with these 3 treatments, what that meant in terms of carbon transfer, what you see here are the data from these 3 treatments, and on the y-axis is the 13C in the roots of the non-labeled plants. So the plants that shouldn't have any 13C in them because they were not labeled themselves. One thing I should tell you that in this box, what Jinho did, he collected those root and soil samples at 2 locations. One location was very close to the labeled plant, like 5 centimeters away, that's close location. And one location was like 15 centimeters on the other side of the box. So, and one other thing I should tell you is this dash orange line indicates, marks what happens with totally unlabeled plants. So that's our non-labeled control. So as you can see, when black-eyed Susan was not fertilized, there was, except for a couple of outliers, there was really any enrichment of its roots with the 13C of that switchgrass origin. When we looked at clover, clover got quite a lot of that. Or maybe I should move to this one. Let me do it this way. So when we look at clover, Clover roots were enriched in 13C of switchgrass origin, both at close locations but even like 15 centimeters away from that switchgrass plant, still there was this an enrichment. So clover was getting carbon from the switchgrass quite a bit. But when we looked at just this black-eyed Susan, which was fertilized, once it got fertilized, things changed quite drastically, because then I guess it became very interesting for the switchgrass to have interactions with that plant. And now we did see quite substantial carbon transfer from switchgrass to this nitrogen-rich black-eyed Susan. So clearly, nitrogen availability is a big driver of why another plant would want to exchange carbon with you or give you carbon. So one not very strong effect, but still what I found very interesting, related to positive effect of highly diverse plant systems in terms of that carbon exchange. So again, this is the one that I just showed you on the previous graph. This is unfertilized black-eyed Susan. And yeah, there is no carbon transfer to it from the switchgrass. But this is our most diverse tree plant species system. And where clover was on one side and that black-eyed Susan across that clover on the other side. And very interesting, having clover as a neighbor with its, high carbon transfer to it from the switchgrass seems to, I don't know, maybe put that switchgrass in a good mood and make it move carbon to the black-eyed Susan as well. Yeah, so again, this is sort of preliminary and not very strong, and just one treatment, but still it's kind of forms interesting future research direction, is that how presence of certain species in diverse community can affect the carbon exchanges and carbon interaction with other species. Okay, so of course, where all this leads us as far as soil carbon goes, and that was very consistent with our hypothesis that the more carbon was exchanged, the more carbon was transferred from one plant to another, the more of it, of that plant-originated carbon were found in the soil. So these are data from our first greenhouse study. On the x-axis is that our measure of carbon transfer, that is what was the enrichment in the non-labeled roots of non-labeled plants. And on the y-axis is enrichment in the soil, or specifically in the study in rhizosphere soil. So in this study, the colors represent these different plant combinations and differing in their quantities of how much was in the soil and how much was transferred. So in both cases, there was significant positive association between what transferred, what is being left behind in the soil. So the next question was then, well, okay, they leave it there in the soil, but can we get at least some idea how long it will stay there? Is it stable? Does it matter how stable it? Does it matter if it's added with transfer, without transfer? Would that affect how stable that carbon might be? So what we did is we took these small intact soil cores from our plots that was done in the first greenhouse study, and then we incubated them. So because they were so small, we only incubated for 10 days. Doing more in them wouldn't make sense. So this is really very short-term assessment of what happens to that added carbon in a very short term. And then also we did in the pairs of those cores, we measured soil 13C before and after incubation. So what we did then was this no-yes groups. We looked at our carbon transfer presence based on the root data, and we separated our treatments into those where there was carbon transfer and those where there was no carbon transfer. So that's what those groups are. And then we looked at our incubation data from that perspective. So what we saw that as far as 13C CO2, that is this losses of that newly added carbon originated from switchgrass or whatever other source plant, in the systems where there was no carbon transfer detected, the losses, the emissions of that CO2 were higher than in the systems where there was carbon transfer. And the same was consistent with just direct measurements of 13C in the soil. The losses were bigger in the plant systems with no carbon exchange, and somewhat smaller when there was carbon exchange. So you might say, well, maybe they already had differences in their organics or something, or maybe that CO2 in general is affected, but actually no, it was only visible, sizable, what the difference between those no-yes systems only was present in relation to this newly added 13 CO2, 13C carbon, was really not there when it came to the overall CO2 emissions from those cores. So our conclusions here would be that at least in that short term, the carbon that is added in the soil, in the systems when there is carbon exchange, it seems to be better protected than the carbon that is added in the soil, in the systems without carbon exchange. So what our explanation is why that could be the case is because probably because of the fungal network contribution to the carbon exchange. So fungi can go into fungal hyphae can go into much smaller pores when they die there and carrying that 13C in them. 13C in those small pores probably will be stronger protected and will stay longer, as opposed to carbon that was just directly deposited into the soil by the roots in bigger pores, will be processed by microbes faster, and losses will be more substantial. So to further explore that, what we did, we actually tried to relate these carbon 13C changes and in the soil. We tried to relate them to pore characteristics directly. So what we did in these, again, intact soil cores, we did this sort of site-specific sampling using a tiny device with set holes. These yellow circles represent those set holes. So in each of the cores, the core was CT scanned, so we knew what, the pore characteristics. Then we inserted that device that gave us soil samples from these specific spots. So we know where the spots are, we have the soil from them, we know what the pore characteristics within each of the spots are. And fortunately for 13C, you really need very tiny quantities of soil, so made it more meaningful as far as relating pores and 13C. So the results were not as promising and conclusive as we hoped, and sort of left more questions than answers. So what you see here are the data for our three plants that So this was done in our first study where we looked at these three specific plants: big blue stem, switchgrass, and wild bergamot. And what you see on the y-axis are these four size groups. And the color scheme represents correlation coefficients between the abundance of pores or volumes of pores in each particular size group and the C in that location. So before incubation, so the results that did make sense and what we were able to interpret were the results from wild bergamot. So it seems what's happening initially before incubation is just what we expected, that there would be more carbon in these medium-sized pores, probably coming from the roots. And then after incubation, so we see these positive correlations, red means positive. We see these positive correlations, but then as after incubation, they change to negative. So seems like what probably happened is that all that carbon deposited by wild bergamot in those pores, it got eaten and lost. But with two grasses, big blue stem and switchgrass, it's actually exactly opposite. And we really found hard time trying to understand why these grass plants would behave differently, what changed in their pore systems or carbon distribution in it that lead to this totally opposite relationship. I don't have, seems like puzzle. So one other thing we looked in our second greenhouse study is we wanted to specifically assess the contribution of the fungal mechanisms to that carbon transfer. So in half of these rhizoboxes, in between here, what's covered by this divider, behind these bars, there is actually just soil, and the roots of the neighbors, they can just freely interchange and grow anywhere they want within the entire rhizobox. So there would be both roots and fungi acting there. And in another half around these dividers, we would put a 53 micron mesh that would still allow fungi to go back and forth, but would prevent direct root contact. So there is this 2 centimeters of space filled with soil, of the same soil, same density, packed the same way, that would separate the neighbors. So the results, this is just a summary of multiple treatments, was just as one would expect, right? That when there was this lots of contact by both roots and fungi, then we have a lot of carbon transfers. But we were surprised that still we had very strong signal about carbon exchanges with fungal only mechanism. So even though there was this 2 centimeters of soil plus the mesh separating the plants, there still were enough carbon transferred from one to another, again indicating importance of that fungal mechanism. So one thing that we keep reminding ourselves that even though here we talk about direct root contact, but probably what we are seeing is not only roots, but also enhanced fungal contact between those roots that are close to each other. So it's really cannot subtract one from another and find root impact, because root means root plus close fungi here. So what does this carbon transfer treatments, what do they mean for soil pores and soil structure? To assess that, what Jinho did, he took intact soil pores from inside of these barriers, so between these rhizobox compartments and did CT scanning and assessed particulate organic matter, and then quantified pores of these different size groups. So what you see here are the volumes of fractions of pores of these different size groups in 3 of the systems with those 3 neighbors, and dark colors represent this barrier when there was only fungi contact, and light is where there was this direct root contact possible. And the orange line is again the one that marks no plant control. And I want to emphasize that these no plant control boxes are the ones they were treated through the whole experiment same as our planted boxes. So we packed them the same way, we put them in the greenhouse the same way, we watered them the same way. So it's just everything was the same except that there was no plants. So when, if you remember the results I mentioned earlier in with black-eyed Susan, there was no carbon transfer. Well, no carbon transfer, no changes in pore characteristics in between the plants. In switchgrass system, there was some carbon transfer, and there were changes in the pore system, especially so specifically what we see is the increase in proportion of these very smaller pores and decrease in larger pores, but that only happened when there was this possibility of direct root contact, so probably was largely root driven. And same was observed in for the clover neighbor, so but was especially pronounced. And very interestingly, even in clover, even when there was a barrier, so again there was this 2 centimeter thing that separated the plants, still I suppose fungal links within that barrier, they affected the pore size distributions and changed pore size distributions there. And that's the one on the black-eyed Susan that was given extra nitrogen, and yeah, there we see same thing. There was quite a lot of changes in the pore size distribution, both because of the roots, but also because of fungal interactions. Okay, so the bottom line is that, you know, that carbon exchanges, they do increase soil carbon and they do affect pore structure. So just one last piece I want to mention, what we did, or what my former PhD student, Ellis did as part of that greenhouse study. She took kind of mini boxes out of our rhizoboxes, and then she mapped enzyme activities on them. So she created maps of these 4 beta-glucosidase, chitinase, and acid and alkaline phosphatases. What you see here is like a sample of her thermography map. Blue is where there was no enzyme activity, and all the way to green and yellow, that's when there was high enzyme activity. So from these, she accounted for like a spatial extent of enzymatically active areas, and for activity, you know, how yellow, green are that enzymatically active areas, how active they actually are. So the thinking was that all this, the interactions among the plants, they would affect not just physically, you know, where root grow, where fungi grow, but they would probably affect the production of enzymes by the plants, production of enzymes by the microbial community. So we did expect to see differences, the impact of our neighbors on them, but we didn't. So it's either maybe the variability was too high or not sensitive enough to detect the differences in the enzyme activities. So we did see some interesting differences in enzyme activities and the extent of enzyme spread among our plant species. We did see differences among soil, so there were interesting things there, but not the neighbors. One interesting result that I do want to mention, because again, it's something that found quite fascinating and puzzling, is the impact of the barrier presence on activity of alkaline phosphatase. So very consistently, this is just a graph from switchgrass, but we saw it in all three plant species, in the bush clover and in black-eyed Susan as well. Very consistently, when there was no barrier between the roots, we saw higher activity in those enzymatically active spots. Activity of alkaline phosphatase was consistently higher than what was there when the interactions were only due to fungi. So keep in mind that that area of contact or no contact, they're right here, and the enzymes were measured inside. So it's not like we even covering that exactly those same areas. So it's just whether or not the plant was sensing that presence of that neighbor or had direct contact with that neighbor. That somehow affected activity of alkaline phosphatase. And what's even more interesting, that unlike three other enzymes, which are produced both by plants and by a variety of microbes, alkaline phosphatase, it's only of microbial origin, plants don't create it. So this presence or absence of direct contact between plant neighbors, somehow miraculously it affected enzyme production by microbial communities under those specific plants. So don't have any specific mechanistic explanations yet, but just found it very fascinating. So in conclusions, what we think this contributes to the understanding of how diverse communities impact soil carbon is that probably when there is that diverse plant communities that is getting started, that interplant transfers of carbon among the community neighbors, enhanced transfers, they are probably what are one of those first steps that gives the boost to soil carbon increases. And again, the more certain neighbors can stimulate carbon transfer toward other neighbors, and importance of close root contact in that process. And so then the more carbon is transferred, the more of it is added to the soil, the more stable, at least so far we know in the short term, that newly added carbon will be in the soil, and the better will be improvements, the more improvements there will be to the soil structure, which again hopefully starts, I'm sure starts this feedback cycle of positive soil organic matter soil structure relationship. So I would want to make sure I acknowledge contribution of everyone who did this work and collaborators and funding sources. And thank you for listening to me, and see if you have any questions.
Any questions for Sasha?
Okay.
I have one. can you go to that last slide, the one with that one? So, so the separation was like separating roots and fungi in one case and then the one case, no? So is that, so seeing like more production of alkaline phosphatase, could it be due to when you have roots, it is creating maybe a little relatively efficient NP zone compared to no, like a nearby root? Could that be the reason? Like, because if you think about why they need to produce that, there should be a reason. Otherwise, they wouldn't spend energy on producing that.
Yeah, that's a very good point. So most likely, yeah, that presence of this barrier or absence of this barrier somehow affected what plants do as far as their phosphorus acquisition, and that made it necessary for them to get more of that alkaline phosphatase out. But what still kind of amazes me is that contact, presence of contact was here, and measurements of the enzymes were here, so they were spatially separated. So it's not like, I would find it much easier to understand and interpret if here there was no roots, so that's what happens with enzymes. But no roots was here, and differences in enzymes were there. So that's to me, and maybe that's just my ignorance about this plant microbial interactions. I still need to, talk closer to people who understand it better. But it's really fascinating how just this plant interaction affects what plant might be doing to its the microbes in its rhizosphere, not necessarily at the point of contact, but in its rhizosphere in general.
And then so it's the carbon transfer, like interplant carbon transfer, and then related to carbon storage or sequestration in pools. It almost seems like, based on what you presented, there's a key player is fungi. Am I right?
Yes.
Yeah.
Yep. And it's very hard. So it's very hard to sort of pull apart the specific contribution. Okay, this is roots, this is fungi.
Yeah.
We tried, but it was bound to fail from the start because it's just we really created this very limiting system with this large distance. So it's definitely we definitely eliminated. So we were able. So basically, with our system, we were able to very conservatively prove the importance of fungi, because even under all these very restrictive conditions that we imposed, they still made this transfer possible in some systems. So my interpretation is that without these very serious restrictions, their contribution probably much bigger. But how do we experimentally tease it out, the specific size? That's, I don't know if it's possible.
I have a question following up like this. When you said you were doing the enzymes, it's more technical question. How you are able to measure each enzyme? Like, was the tomography you did able to do that, or you were doing like the analysis in each spot of the soil? How you were?
No. So this is zymography, basically enzyme mapping. So what you see, this white, it's it's schematic, but that's basically what it was, is that we place a piece of this membrane or sort of fancy filter paper that is saturated with certain substrates specific for each enzyme. So for example, here, this whitish piece of membrane here will be saturated with substrate for beta glucosidase, and we put it on the soil. And well, Melissa did it simultaneously for all four of them, having these separate pieces, like two here, and those are left on the surface of the soil for a certain period of time, like either 30 or 45 minutes, with pictures every minute as the signal develops. And then from that sequence of pictures, then we calculate the enzyme activity. So it's actually not at all, like this might be horrible sounding, sampling, like, well, some of the things we did was this very hard aggregate cutting. No, this is actually quite easy to do.
I had a question related to both your presentations. You highlighted on the pores aspect of like the smaller pores and larger pores. And I think of it as like, is there any relation between pore potential, like matrix potential relationship that could affect some of the interactions that are happening or the exchange that are happening? Like,
yeah, so the way how we do those experiments where we add substrates to large versus small pores, that's exactly based on that relationship or assumed or hypothesized relationship between pore size and magnetic potential. Like that Laplace equation that directly or inversely relates the two. So yeah, that's a underlying principle of that method that should make it work. The reason why there is so a lot of uncertainty is that soil pore system is really not a set of parallel capillaries. It's but yeah, it's right there.
if there is any established relationship between soil texture, like if it varies, like the pores would vary, and then also the potential in those cases would vary, and then does the interplant carbon exchange would also vary?
So as far as this second study, we did this experiment in two soils with not exactly contrasting but different textures and different soil carbon contents and actually different phosphorus availability. Overall, the carbon exchange results were very consistent. Okay. And also these pore size distribution results that we're getting as far as rows of pores of different sizes, they were also consistent in many respects, but now, like in that my yesterday presentation, this last our very recent study where we looked at things where we included soil with very sandy texture. In that sandy soil, things just went crazy in some respects. So. Yeah, and I don't think that's necessarily the physical aspect of it, but more probably mineralogy that contributes to pore in soils of different texture. But that's something that would need more attention.
Thank you. I really enjoyed both your talks. Very informative.
Thanks. I guess I'll have to admit, this is all pretty new to me, and so I kind of got a lot of questions. And I guess the first basic question is, do you know kind of what forms the carbon are that the plant roots are taking up. What is this form of carbon that's being released by one plant root, what's being taken up by the other, and how is, why is the plant, how much carbon is being taken up by the plant root and why? I guess I'll ask that first, and I have two follow-up questions.
So, yeah, I don't know most of the answers.
Okay.
The thing is, and that's, so to begin with, we are not even sure with these carbon results if the plant is, if the plants is, if the recipient plant is actually benefiting from that carbon in a way, or if it's just sort of a side thing. Basically the reflection of AMF fungi in connecting them. So the reason why I'm saying that is that we often, so with many of these recipient plants, we do see a lot of enrichment in the roots, but very rarely, almost never, we see enrichment in the above ground pile. Which means that whatever carbon travels from one plant to another, at least in our study, it didn't really, it stayed in the roots. It never went up. And that is one possible indication that it could be that this is just a vascular mycorrhizal fungi moving from one plant to another and just staying in the roots themselves. And we are not really, we just, what our measurements are, we just take the roots and measure so you can see, so whatever is in the root, fungal, whatever, hyphae and everything, it's lumped together. One explanation, what we think is probably happening here is that what we see as this carbon transfer is just reflecting those fungal interactions. Well, is that what? So what types of carbon compounds that would be that we don't know. In general, plants can take carbon from soluble, easy, available sources with their roots from the soil and use it in their leaves. We've seen that in our 14 radioactive carbon addition study when we, in our soil cores where we added this radioactive glucose, we saw some of the weeds spreading out from the cores, and then we did that radioactivity mapping, and we saw those plants being completely, shining radioactive with the carbon, which they obviously do from the glucose by their roots. So they do that. It's just, I don't think that's what's happening in our case. Another interesting, and again, this is specific to these prairie communities that we studied. Some very fascinating results from literature. So people studied this phenomenon a lot in forests. And one fascinating thing there is what they are finding, where they find a lot of this carbon transfer that take place, where it's not just it's getting to the roots, but where it's actually helping plants is in shaded communities. When like some studies of forest trees would see a mother tree to sort of transfer carbon to those seedlings below ground that do not have access to sunlight. And other studies, they saw similar relationships between plants of different species, different tree species, where a shaded one would be getting carbon into its, using it from the neighbor. So it's really fascinating. So, somebody asked me what I would do differently if I started my career knew I probably would go into plants. But I think in our case, it's really not, they're not using it directly because everyone has plenty of sunshine in the prairie community. But the part that I sort of skipped because I thought that would be too long anyway, is the nitrogen exchange. So I didn't talk about our nitrogen results. So in all these, we also labeled the central plant. We did this foliar labeling with 15N, and then we traced the 15N transfer. Well, that went to, that's 15N, it was in the roots. It was very much whenever there was transfer, the plants used it in their background. So that was, they were using nitrogen, they were taking it purposefully. And with carbon, I think it's just fungal interactions.
Interesting. I think, this goes back to what Congo was saying about the effects of interplant competition on plant response to nutrient deficiencies. And even as you would expect, as there are more roots there would be lower nutrient content and respond with whatever mechanisms they have to acquire more nutrients. And maybe one of those mechanisms would be developing the relationships with fungi or providing more carbon to the fungi, saying, hey, I need your help more, however they do it. And then there's more carbon being provided to that fungi and then it goes out. Or they're saying, more like low molecular weight organic acid release to try and solubilize nutrients. And then those carbon compounds might be absorbed intentionally or unintentionally by the plant roots, just as they're more in the soil. I don't know. It'd be interesting to know what's going. It seems like there could be a strong nutrient competition effect that is driving some of these carbon exchanges.
Yeah. So we certainly saw a positive effect of nitrogen. Nitrogen would reduce interactions, that's for sure. We didn't see, so as I said, like in this second study, we done it in two soils, and one soil was with our usual, fairly high phosphorus available, and the other soil was this calcareous with a lot of carbonates in it and very low phosphorus, and we did think that there would be these, that soil will affect the interactions, that this shortage of phosphorus might be affecting. But the results that we saw, the only thing that happened is that the plants were not growing very well in that phosphorus low soil. But all the relationship, they were there. It's just in poorly grown plants.
I think another problem with something like a calcareous soil, it might not be a good environment for microbes either. So.
Yeah, so that site, even though it does have a very nice soil organic matter level because of all the carbonates, fixing the carbon, but it does have low yields and people have a hard time getting things growing there.
Well, let's go ahead and thank